Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to talk about amino acid classifications. So quick overview, there are 20 proteinogenic amino acids, meaning the amino acids are encoded directly by the genetic code and incorporated into proteins during translation. Each amino acid has a unique R group or side chain that determines its chemical behavior. These side chains allow us to classify amino acids into categories, which is crucial for understanding protein folding, enzyme active sites, and how amino acids behave at different pH levels. We break down amino acid side chain properties into 5 main categories based on polarity and charge at physiological pH, which is 7.4. So let's start with what polarity is. Polarity refers to how a molecule shares its electrons. Some atoms, like oxygen and nitrogen, pull electrons more strongly, which creates a slight charge on different parts of a molecule. This makes the molecule polar, meaning it can mix well with water. In amino acids, polar side chains form hydrogen bonds and like to be near water, while non-polar side chains avoid water and they are usually found tucked inside proteins. So our first category is non-polar or aliphatic amino acids. This just means they're hydrophobic amino acids. These amino acids have a hydrocarbon side chain and they avoid water. They are often found in the interior of folded proteins. Amino acids in this classification are glycine, alanine, valine, leucine, isoleucine, methionine, and proline. In this episode, I'm not going to go over the 3-letter codes and the 1-letter code. That is something that you should study on your own. I'll talk about a game or a quiz you can use after the episode. That might be helpful, but that is just something you should know. Regarding glycine, glycine is unique. It is the smallest and it's not chiral. Methionine contains sulfur, but it's non-polar. And proline has a cyclic structure which introduces kinks in protein chains. The rest of them are just alkyl side chains, which means they have 1 carbon or 2 carbon or 3 carbon side chains. Moving on to the aromatic side chains. These are mostly nonpolar, but they have a varying polarity. These contain a ring structure and can absorb UV light. The largest is tryptophan with a double ring structure, and the smallest is phenylalanine. If you add a hydroxyl group to phenylalanine, you get tyrosine. Now, phenylalanine is relatively nonpolar, but since you add the hydroxyl group to it to make tyrosine, tyrosine is pretty polar. So we have phenylalanine, which is nonpolar, tyrosine, which is the polar hydroxyl group. Tryptophan, which is the largest one, has a mildly polar NH group. So a tip is that tyrosine and tryptophan can participate in hydrogen bonding due to their polar side chains. The next classification we have is polar uncharged. These side chains can form hydrogen bonds, but they don't carry a net charge. So here you have serine, threonine, asparagine, glutamine, and cysteine. Cysteine is important because it can form disulfide bonds with another cysteine. We'll talk about this later. The fourth classification is acidic. These are negatively charged at physiological pH. These have carboxylic acid groups in their side chain, and this consists of aspartic acid and glutamic acid. These are related to asparagine and glutamine respectively. Both aspartic acid and glutamic acid are in their protonated form, which means that their carboxyl R group is COOH. You may also see their deprotonated forms, which are aspartate and glutamate, and their carboxyl R group is COO-. The difference between aspartic acid and glutamic acid and asparagine and glutamine is that asparagine and glutamines have amides in their R groups, whereas aspartate and glutamate have the carboxyl groups with the COO-, giving them the negative charge. Finally, the fifth classification is basic amino acids. These are positively charged at physiological pH, and they have side chains with amino groups that accept protons. Here you have lysine, which has a terminal amino group, arginine, which has 3 nitrogen atoms, which is called the guanidino group, and the positive charge is delocalized through the 3 nitrogen atoms. This is also strongly basic. And you have histidine. This has an imidazole ring, which has 2 nitrogens on an aromatic ring. It can be charged or neutral at physiological pH. Now this is important because its side chain has a pKa of around 6.0, so it's found in enzyme active sites where it can act as a proton donor or acceptor. It does so at physiological pH when one nitrogen atom is protonated and the other isn't. Under more acidic pH, the other can also become protonated, giving it a positive charge. For amino acids, I generally think you should make your own mnemonics. Whatever resonates with you would probably be better in helping you remember them. There's also this app on the App Store called the Amino Acid Quiz. It's free. It quizzes you on 1-letter, 3-letter abbreviations, structure, name, and you can test yourself based on any combination of those things. So you can use the 1-letter abbreviation to choose the structure, or you can get the name to choose a 3-letter code. And I think it's really helpful if you do that a bunch. Just by doing that, you should be good. But if you want a mnemonic to remember the different classifications, I would suggest not remembering the non-polar and just remembering that if it's not in the other ones, it's non-polar. For charged, you can remember dragons eat knights riding horses. So you have D, which is aspartic acid, E, which is glutamic acid, knights, or for K is lysine, R is arginine, and H is histidine. Then you can remember your polar uncharged. So Santa's team crafts new quilts. So serine, threonine, cysteine, asparagine, and glutamine. You would know that everything else is either an aromatic or non-polar. And there's just 3 aromatics. You can remember that by wifey, or you just, of wifey, if you spell it out, it's W-I-F-E-Y. And just know that W, F, and Y are aromatics. So tryptophan, phenylalanine, and tyrosine. And if you just remember their polarities or non-polarities, then you'll know that everything else is non-polar. But I would recommend using that app, and I think you should be fine. So overall, some conclusions we can draw are that amino acids with long alkyl side chains, like alanine, isoleucine, leucine, valine, and phenylalanine, are all strongly hydrophobic and are more likely to be found in the interior of a protein away from water. Amino acids with charged side chains, positively or negatively, are polar and hydrophilic, as are the amides asparagine and glutamine. The remaining amino acids lie somewhere in the middle, neither particularly hydrophilic or hydrophobic. So to wrap up, understanding amino acid classifications helps with predicting protein structure and solubility, recognizing functionally important residues and enzymes, and anticipating pKa shifts and interaction types in biochemistry questions. In the next episode, we'll look at how amino acids act as acids and bases, focusing on pKa, titration curves, and buffering.